afternoon, everybody. And I'm Rosemary Collier from Warwick Crop Centre, the one who has been emailing you. So thank you very much for joining our webinar on the future of vegetable crop breeding. It's quite timely because the importance of genetic improvement and crop breeding was highlighted in the recent government blueprint for the UK fruit and vegetable sector. And certainly as a researcher working on pest insects, I see pest resistant crops as a key tool for the future. So the webinar will consider the opportunities and technologies available for breeding new varieties of vegetables in the future. And there'll be short presentations by Dr Eleri Tudor and Dr Justin Roberts, both from Elsoms Seeds, and Dr Lauren Chappell from Warwick Crop Centre. So just to flag up that we'll be recording the session and it will be available later, and I'll send out the links. I've applied for basis points. We'll take questions at the end of the presentations, but feel free to type questions in as we go along. My colleague Alex Kelly set up this system, so I'm not sure whether you're allowed to speak or not, so we might have to take typed in questions when we get to that if you can't actually speak to us as the audience. Right, so I'll stop sharing and I'll hand over to Justin and Eleri first. So Justin's going to speak first, then Eleri, and then Lauren. So thank you all very much.
Thank you, Rosemary.
Great, slides looking good.
Brilliant. Okay. Yeah, I just want to say thank you to Warwick for giving us this opportunity to promote what we do here at Elsoms in vegetable breeding. It's greatly appreciated. Okay, so just wanted to give everybody a little brief overview of who I am and what my background is. I've been at Elsoms for almost 7 years now. My main responsibilities are for the veg, herb and leafy salad breeding on-bred crops here at Elsoms. I joined the company in 2017. I was midway through my PhD at Harper Adams at that point, and I completed my BSc and MSc prior to that at the University of Salford. So my main responsibilities and our on-bred portfolio here at Elsoms have a wide range of species, from the sprout and broccoli and spring cabbage in the brassicas, turnip and swede, parsnips, but also we do herbs, so we do basil, dill, coriander, parsley, and also some leafy salads such as wild rocket, chard, cutting celery. We not only just distribute our own bred varieties, we work closely with international partners such as Bayer. We distribute many other vegetable varieties here in the UK. We have a seed technology department here in the UK that specialises in various seed treatments and seed priming. But Elsoms also have other breeding programmes with collaborations. So we have a partnership with Sarton Union for Elsoms wheat and also with Ackman for Elsoms Ackman barley. Elsoms has continued to grow in the relatively short time that I've been here. So recently started up Elsoms Trees for the UK distribution of tree saplings. But also even more recently, we've established Elsoms International, which is a partnership with Crookham's and Crite Seeds for the Europe-wide distribution of sweet corn and peas. Before I go into what I think of the future of breeding, veg breeding is, I'd like to give a little bit of an overview of what we currently do at Elsoms. So what we do is, for some species, it's still underpinned really by conventional breeding methods. So single seed descent, I won't go too much into that because I know Larry's going to touch on it. But we're moving more and more of our species toward produced material through anticulture and microspore culture, DH production. And we're now using marker-assisted selection and marker-assisted recurring back crosses in most, if not all, of the crops that I'm responsible for. And the spread breeding technologies, we use that generally for any back crossing and marker-assisted work, just really to get through the generations as quickly as we can and get those traits that we've got the markers for into our elite material. We have undertaken mutation breeding before in the past and still do. This is through EMS treated seed, and that's basically we use that to try and create some variation within the species. Okay, so the next few slides is where I think we'll be heading at Elsoms in the future, or what I think we should be looking into. As I said, double haploid technology. That would be great if we had that in all species. There's two aspects for us really as to why that would be a good thing. The first is it gets to new improved material to grow sooner. As you can see from the diagram, the speed it takes to do conventional single seed descent and the benefits of double haploid production. Double haploid production is a little bit more shotgun than it is to do conventional breeding, where you've identified and following the trait through the generations, but it dramatically reduces our costs of taking through all the generations and speeds that up. Currently, though, we don't have the ability to produce double haploids in all species. Some are recalcitrant species, and some of them have low returns for the effort that we put in. But yeah, the focus in the future for us would, if we cut the costs on breed side, would be to produce everything through double haploid. Hybrids. One of our aims would be hybrids for all our species that we work in. Hybrids allow us to provide uniform crops without losing the vigour through inbreeding depression, which is the heterosis. But also it can allow us to combine genetic traits from two parental lines without the need for stacking them in open pollinated lines. We currently use two methods at Elsoms for producing hybrids. So historically, we've used the self-incompatibility alleles from the Brassica oleracea, but they can be leaky and you can end up with some inbred parental material in the hybrid crop. So now for all our F1s that we produce, we use male sterility. That gets us around the leakiness of the self-incompatibility, but it also allows us to protect our material. And one of the reasons we want to protect our IP is breeding programs are very expensive. They're expensive to run, so it's important for seed companies such as Elsoms to be able to protect what we release and to try and ensure that we get a good profit from those investments. And the reason this slide's up there is because we don't have F1s in all species. Male sterility doesn't exist in some, but challenging it, developing it can be very challenging. Developing male sterility will require either protoplast fusion or wide species crossing. And this is, I say, this can be challenging. High risk because it's not guaranteed to work, and that work would also inflate the breeding program costs. So yeah, challenging traits. This needs to me to where vegging comes involved. So vegging is a really important research group for the species that we breed in. The truth is that most vegetable species just don't have the budget that the cereals and the big crops such as even carrot and lettuce and onion, for instance, do have. So what's important for us is that vegging listens to what the UK breeding companies' needs are and focuses on what will drive these undervalued species forward in the UK. For me, vegging also has the scope to look at traits that we don't necessarily see as directly impacting value of the seed for either us or growers, but do have consumer importance such as taste and nutrients. nutrition, for instance. Another important part for us is germplasm and where we can get the genetic resources. So gene banks for us are really important sources. Whether that be for us identifying novel traits and new sources of resistance through cultivar accessions or wild relatives. But in my opinion, gene banks are underfunded, and many of the accessions that we access are not yet characterised, and take a lot of our work just looking at the different accessions to try and find what we need. Breeding for the future, phenotyping, large bottleneck for us is phenotyping. Phenotype is an essential part of what we do. Each vegetable species has different target traits that require phenotyping. Therefore, automation can not only be costly to develop, but can be needed to be made bespoke for each individual trait. Some traits we work with require manual input anyway, such as root crops. You've got to lift them out of the ground and wash them, and other internal destructive testing as well. So, for instance, brown height and Swede. Each market population we produce is usually aimed for a different trait of interest. So trying to automate these and bespoke phenotyping for these traits is costly, and for the sake of doing 2 or 3 tests on them populations, it doesn't really sometimes stack up. So most of our phenotyping we usually do is still manual, and I see it still being manual, and which can be very time consuming for us. Other things that I think are interesting for the future are varieties for automated harvesting. This will reduce the issues in the future associated with things like the labour shortages we've seen, or at least reduce costs from multiple harvests. For one example of this is sprouting broccoli. So traditionally, sprouting broccoli was a bush type plant where the growers would have to pay people to go through multiple times and manual harvest this crop. An example of how we developed that is Burgundy, for instance, which is one step from those original varieties, and that the harvest is still manual, but at least it's one pass. All the spears are ready at one time. As automated harvesting becomes more and more prominent, future challenges will be to ensure varieties can stand up to this, making sure that the morphology of the plant is right for the automation, etc., etc. Another one I've added in is more of a personal one, is new product development. So we get often asked from different groups to drive new product development or develop other novel crops that are not necessarily the norm in the UK food consumer diet. But without the drive and the willingness to change this from a consumer point of view, it's whether we can actually justify this as a breeding company. We can't talk about breeding for the future without mentioning climate change, obviously. This can be a difficult one for breeders in the future, as I don't think we're going to be solely breeding for a single change for extreme. So you can't solely breed for waterlogging tolerance or drought tolerance, or even increased pest pressure that we're likely to see from the winter months warming. We need to take into account extreme swings, where we are already seeing trials experience both waterlogging and drought, or extreme shifts from minus temperatures to well over 12, 13 degrees in a matter of days. Yeah, just as a point to the image, I'm not quite sure it's that clear, but the image in the background was a trial from early 2023 that suffered just that. So it went from -3 or 4 right up to 12, 13, even 15 degrees. And the trial, most of the plots in the trial didn't stack up too good towards it. We need to start breeding for resilience for multiple climate change factors, I think. Conventional breeding allows us to select the material as it did that year, and it does work quite well. But we also need to start to identify the genes that make resilience possible, which is where Eleri comes in. And that's over to you.
Okay, thanks, Justin. So my turn. So I'm Eleri. Great to speak to you this afternoon, and I'm the Genomics Specialist here at Elsoms. And my role here is to lead the molecular biology activities to support the plant breeding programs and also our seed production operations. So I'll start my half of the talk by introducing me and where I've come from. I was born and raised in South Wales and went to the University of Oxford to study biological sciences. And during my final year, we did a module on food security. And I really think that was the catalyst for me. You know, it drove me to pursue a career in sustainable agriculture and plant genetics. And I came to the University of East Anglia in Norwich to study a master's in just that. So I did a master's in plant genetics and crop improvement, and then hopped over the road to the John Innes Centre to complete my PhD, looking at the genetic control of flowering time in brassica species. And then back in 2019, so nearly 5 years ago, I came here to Elsoms, and I've been here since then. So Justin has outlined a lot of what goes into plant breeding, but I thought I'd talk a little bit about the plant biotechnology side of plant breeding. So we can use things like, or tools like genetics, genomics, marker-assisted selection, genomic prediction, gene editing, and genetic modification to develop new and improved varieties. And using these plant biotechnologies, we can identify the genes that are responsible for a trait of interest. We can determine how they work in the plant. We can select for those genes in a plant breeding program and transfer those genes into plants where they are needed. So as Justin alluded to, conventional plant breeding takes time. And generally speaking, it relies heavily on visual selection in the field. So a plant breeder will go out and look at his or her material in the field and select the best of that material to go forward into a crossing program. So if I just outline a very theoretical example of what a conventional plant breeding program looks like. So let's say we identify a disease-resistant line. However, agronomically speaking, it doesn't do very well. So perhaps it doesn't germinate very well. It has low yield. Maybe it's susceptible to lodging. So in a plant breeding program, what we want to do is take that disease-resistant trait and cross it into our elite cultivar or our elite material, which I've represented in green. So what we'll do in a plant breeding program is cross that disease-resistant material back into our elite cultivar over and over and over again, continuously at each generation, selecting for that disease resistance, but in the background of our elite material. However, there are downsides to relying on just purely on visual selection, because some traits are dependent on the environment. So, for example, you can't assess for disease resistance if that disease isn't present in the environment that you're growing your material in. Some traits are not visible. How do you visually select for those? And then some traits are expressed late in development. So yield, for example, you have to wait a long time, until the plant is mature and setting seed before you can select for those lines that are performing the best. So we use, and many plant breeding companies use, marker-assisted selection as an indirect selection process. So this tool allows us to use DNA sequence information or DNA markers as a proxy for those traits that we're interested in. So if I go back to my theoretical example, in yellow is my disease-resistant plant, and in green is our elite cultivar. But this time we know we have some information on their genetics and their DNA sequence, and we have identified the disease, the gene that underlies that disease resistance, and know where it's located in the genome. So instead of visually selecting for the disease resistance, we can screen the offspring for the presence or absence of that disease resistant DNA marker or gene at each generation. So at the seedling stage, we can look at the DNA sequence in our material and select those individuals that carry the disease resistant trait. by looking for that DNA marker and then get rid of any plant material that is likely to be susceptible. So in a plant breeding program, we can use this tool to look at single markers or genome-wide markers. And the sole aim, so the real aim of this is to identify our genetic best individuals in a population. And we can use this as a tool to track the favourable traits over generations so that we don't lose that trait. We can select for traits that are difficult to measure and also remove undesirable traits. So plant breeders now have a huge amount of genetic and genomic resources available to them because hundreds of plant genome sequences are now publicly available. Also, with the advancement in genotyping technologies that are available, us as plant breeders, we can now screen hundreds, if not thousands, of samples per day for genetic variants. So I'm sure many of you have seen these kinds of graphs before, just showing the amount of data that can be produced in a day nowadays. I mean, this plot is quite old, but compared to 20 or 30 years ago, with the evolution of next generation sequencing, but also with that comes a reduction in the cost of producing that data. So nowadays, and I'm sure going forward in the future, we are able to produce loads of data at significantly less cost. So we're now using, and I'm sure we will use in the future, this genotypic and genomic data to make more informed decisions in our plant breeding programs. And ultimately, it saves space because we can remove any material that is poor performing, but also saves time and money. But there are other ways to introduce. So what I've spoken about so far is how we identify and screen for the diversity that already exists in our plant breeding programs. But there are other ways to alter genetics in a plant breeding program. I'm sure many of you are aware of these. So genetic modification is one tool that enables plant breeders to improve crops in ways that would not be possible by other methods. So this is a tool that allows us to move genes from one species to another, which isn't possible without genetic modification. However, in the UK and Europe, GM is strictly regulated, so it's not something that we are using at Elsoms. But we can also, Justin mentioned mutation breeding, we can use chemical, biological and physical mutagens to induce genetic variation in plants or in our material to alter the traits that we're interested in. But this is quite a random approach. It causes these mutagens cause random mutations across the genome, so you don't always end up with a predictable phenotype. But in contrast to GM genetic modification tools, this method is it's exempt from GM regulation, so we can freely use these kinds of tools for inducing genetic variation. And then lastly, but not least, is gene editing. And in Europe, obviously, gene editing is currently strictly regulated, but public and political perception are changing. And obviously, with the Precision Breeding Act in the UK, we might be able to use gene editing more freely in the future, but it's all obviously under discussion at the moment. But what does all of this? How does all of this impact Elsoms as a relatively small UK independent plant breeding company? So, as Justin mentioned, we have a huge vegetable portfolio, and this slide just represents the vegetables that we sell and distribute across, like, globally. And some of this is our own bred material. However, not all crops have genome sequences. Not all crops have population genomics data, so we don't know the genetic diversity within them. Not all crops have markers associated with agronomic traits. And then also not all crops can be transformed to make them GM or gene edited. So if we just take the Apiaceae family of crops as an example, so we have a few APAC crops that we breed and distribute. These include things like parsnip, carrot, and coriander. And so this is the information that we have for these species out there in the public domain. So if we take carrot as an example, carrot has a reference genome sequence. There's population genomics data, so we can assess the genetic diversity within carrot. There are published markers for traits, and it's easily transformable, so you can make GM and GE carrot. Celeriac, parsley, and parsnip, in contrast, don't have any public data available for these kinds of research areas. So, you know, we don't have a genome sequence for celeriac, parsley, and parsnip in the public domain. Coriander, celery, and dill, there's a bit of information out there that we can use. Some of it is useful, some of it's not so useful, but the APAC especially is they're very much an under-researched crop. So because of that, Elsoms have to, if we want to do any research in these areas, in these crops, we have to do it ourselves, or we have to do it in collaboration with a academic research institute. So, for example, we currently have a PhD student with the Elam Institute, Charles Dickinson. He's currently working on coriander, and part of his project is to develop some population genomics tools so that we can assess the genetic diversity within coriander, and in future develop markers for traits that we're interested in. And previously we've had a collaboration with the University of Warwick to develop genomics tools in parsnip. So to summarize, plant breeding has been extremely successful. If we take wheat, for example, the average UK wheat yield has almost quadrupled over the last century. And I'm sure within the vegetable growing and breeding world, you know, we've seen similar increases in yield. Part of this has come from, you know, improvements in agronomy. So we've seen improvements and developments in mechanization, development of artificial fertilizers, use of pesticides and herbicides. These have all made significant improvements to crop yield. And plant breeding has also provided an important contribution, but I would say mostly without the use of plant biotechnology, especially in UK vegetable breeding. But now and going forward, there's a huge amount of genetic information and resources available to us from the genomics era, and will be produced going forward, I'm sure. This amount of information and resource greatly exceeds what has previously been available to previous generations of plant breeders, so that they could work with. So thank you very much for your listening, and I think Justin and I are pleased to take questions at the end.
Brilliant. Many thanks, Justin and Eleri. So I think we'll probably take the questions from your presentation, if that's okay, and then we'll go on to Lauren. So I may have confused everybody, because in this version of Teams you can do Q&A as well. So in the Q&A, Richard Crowhurst asked, for the non-geneticists among us, could you just explain the principle of double haploid breeding compared to conventional crossing?
Sure. So double haploid is a technique that we use in the lab. It's a tool really to fix. We use it to fix the genetics. Essentially, it's so we use agricultural microspore culture, and we take cells from the anthers that would ordinarily produce pollen, but we switched their developmental trajectory, I guess, so that they produce embryos instead. So the haploid, cells in the anthers are haploid, and then they spontaneously double in the embryos. so that you capture all that the genetics that were in the haploid cells of the anthers, but then they're spontaneously doubled, so they're genetically fixed. Hopefully that answers the question.
Thank you. Come back, Richard, if you would like more. And then one in the chat, which is from Henriette, which is, why can some crops not be transformed or gene edited?
It's a very good question. I don't know, but there is so how amenable a crop is to regeneration and transformation is species specific, but also line specific as well within a species. So some species are really amenable, and some species aren't, and I don't know why. But there must be some kind of genetic reason for it.
Great, thank you. I think that's the two questions for the moment. Okay, so we'll now thank you both again, and we'll now hand over to Lauren Chappell. So Lauren is a key player in the Vegetable Genetic Improvement Network, and that's a project that is hosted by University of Warwick Crop Centre and Harper Adams University, and funded by DEFRA. So over to you, Lauren.
Thank you very much. Hopefully everybody can hear me and see the slides.
Slides,
yes.
Yeah, fine. Slides fine. Sorry.
Brilliant. Thank you. Yes, as Rosemary said, I'm going to be talking today about our VEGIN project, which is our Vegetable Genetic Improvement Network. So VEGIN is funded by DEFRA. It's currently its 10th year of funding. And DEFRA is all about this kind of interactive network of researchers and industry leaders working together to promote market delivery of improved vegetable varieties using sustainable production systems. It's a really collaborative project. We work really closely with industry, including breeding companies such as Elsoms, agronomists, growers, everybody within the industry, to really try and identify and improve vegetable crops. We work on 4 key UK vegetable crops currently, so carrot, brassicas, lettuce and onions. I should say vegetable brassicas. And the project is kind of primarily divided into 2 main themes. So abiotic, which is that kind of climate-based research that Justin was talking about earlier. So that drought resistance, heat resistance, salt resistance, all of that. And that work is primarily carried out by Harper Adams University. Here at Warwick, we focus more on the biotic traits. So they are primarily the pest and disease resistance. So VEGIN really focuses on kind of developing these genetic resources and tools to accelerate breeding for sustainability traits in vegetables. And we do this through establishing this kind of network with industry. We really work to improve knowledge transfer amongst industry, but we work really closely with the Genetic Resource Unit, but UK Vegetable Gene Bank, based here at Warwick. So the UK Vegetable Gene Bank are all about conserving and using vegetable crop diversity. They hold thousands of accessions, so they currently hold over 14,000 accessions of over 47 different vegetable and herb crops. And this seed originates from over 100 different countries. And this is really important for us because this is the genetic diversity that we're talking about and we're utilising. We're trying to utilise this genetic diversity to identify and understand these novel crops that will be linked to these key traits, these key improvement traits that we want in our vegetable crops. So I said in VEGIN, we focus on the vegetable brassicas, onions, carrot and lettuce. And hopefully you can see from this table that the UK Gene Bank contains thousands of accessions for each of these. 1,500 to 5,000 is a huge number of accessions to be working with. So within VEGIN, we curated diversity sets. And these diversity sets represent the diversity held by the UK Vegetable Gene Bank, but in a kind of more manageable level. So we typically have about 100 accessions in our diversity sets. And why is this diversity so important? Well, if you kind of think of our traditional wild type vegetable crops, they had a huge mixture of genes in them. If you kind of, it's almost a bit like a funnel system. You start off with loads and loads of genes, but it's hugely diverse, and you wouldn't even recognize many of them as being vegetables. Through selection process, we've kind of got these land races, which are kind of these traditional accessions that have been found regionally throughout the world. And that was the kind of domestication step. You started funneling down those genes and selecting for certain genes, and you start to see some uniformity in those crops. For example, perhaps the same color, or they're starting to look the same shape. However, that's not really suitable for commercial environment. So this is where kind of this modern plant breeding has come in that Justin and Larry were previously talking about. And this has enabled us to get kind of really uniform crops with very specific traits, perfect for kind of UK horticulture. However, what this does mean is we're kind of narrowing down those genes even further. We're fixing those genes so that we get really good crops. However, we're limiting that diversity. So in vegging, we need to go back and look at these wild types, these land races, and really utilize these diverse genes that are available in them. So as I said, we have these diversity sets that we've curated from the UK Vegetable Gene Bank. There are approximately 100 accessions in each of our diversity sets for each of our key crops, and they really encompass the kind of variation that you see within these crops. So they're all different shapes and colours, they're from all over the world, and they all have different kind of characteristics. And the idea is that that should represent all the genetic diversity within those crops. So this is fantastic because within vegging, we work on quite a huge number of traits actually, and I'm just going to briefly introduce some of our diversity sets and the current traits that we're working on. So in lettuce, hopefully you can see our diversity set in the middle there, all different shapes and sizes. And within lettuce, we're currently looking at fusarium resistance. So fusarium is a soil-borne pathogen, causes major vascular wilt and root rot. We're looking at lettuce pinking and browning, which is primarily a post-harvest problem. Again, we're looking at that droughting and flood tolerance. Drought tolerance in lettuce is particularly important, particularly the summers we've had the last couple of years. And on the insect side, we're looking at resistance to current lettuce aphid. In our vegetable brassicas, again, we've got a couple of different pathogens. Club root causes this clubbing in the soil, reduced yield. You can experience fairly good tolerance in some brassicas, but it does cause major yield losses. And on the insect side, we're focusing on things like diamondback moth and cabbage root fly. In carrot, one of the major pathogen problems currently in the UK is cavity spot caused by the Uomycete Pythium violae. Again, carrots are very susceptible to drought and flooding tolerance, so Harper Adams are looking at that. And we've recently started looking at willow carrot aphid, which causes yield loss in carrots, but actually the more significant problem is it causes, or it rather transmits, carrot red leaf virus, which causes kind of reddening on the leaves, necrosis in the carrots, and causes major yield losses. And of course, onion. So in onion, we're looking at things like bean seed fly. Bean seed fly causes major damage to germinating seedlings. You'll often find that you'll never see any germination in the field because the bean seed fly have nobbled them all. Onions are, again, very susceptible to climate change. So things like drought and flooding are being looked at Harper Adams. And we're also starting to think about things like onion thrip. So what is the kind of pipeline that we have in vegging? Our kind of first steps really are the kind of genotyping and phenotyping. We use those diversity sets that I previously described, and we develop specific screening assays for the trait that we're focusing on. Our traits are decided very much in collaboration with industry. We have regular conversations with stakeholders to understand the key problems that they're experiencing, and that's how we would decide our programme. And then we start developing these kind of resistance screening assays. And we can do that in kind of controlled environment conditions, in the glasshouse or out in the field, whichever is appropriate. More often than not, we'll do multiple screens in multiple locations. And typically for our diversity sets, you'll see a graph similar to the one we show here, which is segregating for the trait. Because we have such a huge range of diversity in the genetics, you see a huge range of results. which is fantastic news for us because that typically means we'll find some accessions that are more resistant or more tolerant and some that are more susceptible. And once we've kind of confirmed this through multiple testings and research, we can then start to perhaps look at carrying out genotyping assays. So this is very much kind of some of what Eleri was talking about earlier. We'll use existing data that we may have, utilise new technologies, new genomics capabilities to start to identify the gene of interest or the trait of interest, try and identify where it is and what it is, carry out genetic mapping. At this point, we often need to do crosses to get segregating populations. It depends on the technology and the resources that we're using. But at this point, the whole aim of vegging is to try and understand those genes of interest. Vegging is very much about pre-breeding. Our aim is to develop the assays, understand how these pests and pathogens work, identify the trait of interest, identify the gene, and at that point, once we know where it is and what it is, we can hand that information over to the breeding companies for them to carry out their breeding process into their elite lines. So we very much do all that pre-breeding step, and the actual breeding we leave to the experts, the breeders like Justin and Eleri. So I thought I'd give you a little example now of some of the work that we've been doing in that kind of pipeline with willow carrot aphid. So willow carrot aphid is, as the name suggests, a pest of carrot crops in the UK. It overwinters on willow and will migrate to carrot crops in the spring. As well as carrot crops, wild Apiaceae are also a reservoir for this pest. So things like cowparsley and hogweed, you'll see lots of them around us at the moment. And these aphids themselves will feed on the plants and cause yield losses. As I previously mentioned, the more significant problem is that they will transmit viruses. The most significantly important of those is the motley dwarf complex, and within that the carrot red leaf virus, which, as the name suggests, causes reddening of leaves, but significant yield losses by kind of root necrosis. So we really need to start to understand this pathogen, or this pest rather, and look for resistance. So we started off by sowing out our diversity set into the field. So these were our kind of 96 accessions. It was drilled early April, and aphids were counted in the field to coincide with the first migration. And hopefully you can see here, this is just a subset of our data to make it easy to look at, but you'll see there a nice range of aphid numbers on the plants. We've got that segregation that we're looking for. And number 7 that's highlighted in the middle there is Nairobi. Those of you that are familiar with carrot crops will know Nairobi to be perhaps kind of the industry standard carrot at the moment. So that's sitting nicely in the middle, maybe showing some tolerance, but certainly not super susceptible. So this was really interesting. This means we've got some accessions that are resistant, potentially, some accessions showing more susceptibility. So at this point, we wanted to move this into more controlled environment conditions. So we set up pot trials within our insect rearing unit, and again looked at infesting pots with willow carrot aphid and then counting the remaining aphids after a set number of days. And again, you can see here we've got some segregating data. It is to a lesser extent. Our Nairobi accession, number 7, is still slap bang in the middle, which is really good news. And whilst there are subtle changes between those accessions, those that are resistant are still resistant, and those that are susceptible are still susceptible. So at this point, we really want to look at testing this a bit further. Working with insects is really challenging because of the kind of modes of resistance that are present for insects. And essentially, insects kind of have free will to a certain extent. So if the choice is make do on a plant or die, they typically make do, which you wouldn't necessarily always see in a field environment. So at this point, we developed some choice experiments to allow the aphids to choose whether they would rather go on variety A or variety B. And in these choices, we typically gave them Nairobi versus, in the first set of experiments were some commercial lines currently available, Norwich, Norfolk and Nazareth, which are actually all Elsoms lines. And in our second kind of set of experiments, we looked at Nairobi versus some resistant accessions identified in our previous screens and some susceptible accessions. So the setup here is kind of the opposite accessions in opposite corners, and then we release winged aphids into the middle and they can choose where they go. And the results from this were really interesting. So on the left-hand side here, you can see our commercial line experiment. And here, all we're really seeing is that there's not much difference in aphid choice between our commercial accessions. Perhaps a slight preference for Nairobi in some areas over others. So meaning those elite cultivars that have been more recently bred are perhaps slightly more tolerant to aphids. But what's more exciting is perhaps our diversity set choice experiments. So you can see here in blue, we've got Nairobi, and you can see Nairobi versus accession number 8 and Nairobi versus accession number 3, which are our 2 resistant accessions. Not a single aphid chose to go on those resistant accessions compared to Nairobi. And if we look at that in comparison to those 2 susceptible accessions that we identified, accession 10 and accession 4, there were aphids choosing to go on those susceptible plants. So for us, this is really good news. We've carried out some initial field screening, which identified resistant accessions. We've managed to continue that in more controlled environment conditions. And by then testing that further in a choice experiment, those resistant accessions at this point are really holding up. So for us, the next stage for this is to carry out that kind of genetic analysis that Eleri was talking about. We want to start understanding what's causing this resistance, what the genes or traits responsible are, and can we identify what they are and take that forward. So kind of, I guess, a brief summary of Vegin and what we do. Vegin really aims to identify improvement traits in key vegetable crops. We collaborate closely with industry, and we've kind of developed this pre-breeding pipeline to share novel genetics with breeding companies. In terms of the willow carrot aphid, willow carrot aphid is definitely a major problem for carrot production in the UK. And we've developed some really nice field and control environments resistance screening assays with our carrot diversity set. By further kind of scrutinizing the accessions we've identified, we've definitely kind of got some really clear resistant accessions that we are now taking forward to try and understand what the basis of that resistance is. As I mentioned, this willow carrot aphid transmits the viruses, so we're also starting to look at virus diagnostics. We're in the process of optimizing a LAMP assay so that we can identify rapidly the virus, and we're hoping to then start moving on to looking at virus resistance. Is this the same as the insect resistance? Probably not. It's a very likely to be a challenging story. I just wanted to end highlighting kind of what Rosemary started with, actually, is this blueprint paper that the government released a couple of weeks ago, and just really highlight that we're at a really exciting time. You know, we do have a great need for improving our vegetable crops. It's a very challenging time for growers and breeders and the whole industry. But actually, I think we're in a really, really positive place that it looks like additional funding and interest is really pushing in this area. So maintaining these close collaborations, continuing to work together, I think it should be for a really positive environment. Just going to end up by thanking a huge team on Vegin. There's lots and lots of academics. So thank you all so much. And thank you to DEFRA funding. And I'd be very happy to take any questions.
Excellent. Many thanks, Lauren. Right, so we've got a few questions, some of which I think probably are directed or better answered by Eleri and Justin. So I'll try and do them in a logical order. So I think definitely one for the Elsoms team is from Chris, which is seed production is becoming quite difficult with the climate issues. What are your thoughts on this rebreeding?
Yeah, it's definitely something we're looking at. We've just had a PhD with Cranfield looking at flowering architecture in parsnip, for instance, to try and increase the algae production in parsnip. As climate change kicks in more and more, whether we will then have to move where we do our productions to different areas is also an option for us. But yeah, it's definitely something we're looking at for the future. I hope that helps.
Yeah, thank you. Right, and then there's one from Gary, which is probably yours as well. Can you say something on creating interspecific crosses between different species of very similar vegetables? Is it more possible with new techniques? I'm thinking of where one species is highly susceptible to a pathogen, but the other species shows strong resistance. Maybe a cross could be very useful. Thank you.
Yeah, so we do play around with some embryo rescue for wild hybrid crosses. It depends how far apart the species are, I think, to how successful you are. But you could also use bridge embryo rescue, where you do hybrid bridging, where you cross one with a closer a species that's similar to both, in the middle of both historically, and then move it across that way. That there's a lot of work involved in wide hybrid crossing, so you've got to be sure what you're going for.
Right, thank you. And another one, this one's for Eleri from Emma. How stable are new traits in genetically edited crops? Are they fixed, permanent changes?
Ooh, yeah, I guess it depends on the edit. So I'm not a gene editing expert for sure. Maybe someone else can answer it better than I can, but I think it all depends on how you've edited it and what the edit is, I think, whether it's heritable or not, I should imagine.
Right, I don't know whether if anybody's wants to add to that, please put it in the chat. That would be good. Thank you. Thanks, Larry. So then we're on to the willow carrot aphid experiments. So first of all, one from Emma. So Lauren, how repeatable are your willow carrot aphid choice experiments with the diversity set and commercial varieties, given low levels of aphid, 0.5 aphids per plant in the experiment?
Yes, it's a really interesting question. Actually, this one was fairly repeatable. So the choice experiments in particular, they were repeated, I think minimum was 6 times, some of them 10 times. So actually that they are fairly reproducible results. We're aware that if we perhaps change climate conditions slightly, the aphids may make slightly different choices, which is something we're starting to look at now. But certainly in terms of that kind of resistant, non-resistant, susceptible choice, that's very reproducible.
Thank you. Right, so then one from Andy. Very interesting talk, Lauren. Can I ask about the willow carrot aphid resistance screening? Why was the manual infestation experiment assessed after only 5 days? Was the trial not continued further to determine reproduction on the carrots too, and the development rate of the aphids on the varieties?
Yes, again, great question. So we assessed after 5 days because at that point the aphids had produced babies, if you like. So they completed their life cycle on the plants. So we knew that they were able to feed and reproduce. There were, as this is in control environments, I have to say carrots don't do brilliantly in control environments. So that's one of the reasons why we don't continue the assay, because the carrots themselves, they don't do brilliantly under control environment conditions. So at that point, you're not sure whether it's the insect that's having an effect or just because carrots would much rather be in the field, to be honest. But in terms of development rate, that's a very good point and something we're hoping to look at.
But I think that there is another aspect to that, in that we're actually interested in virus transmission, which is, I guess, mainly by the winged aphids arriving in the crop. So how important the next generation is in terms of virus transmission, I don't really know. So I think that's another reason for doing the choice by the winged aphids. So more questions. So from Richard to Lauren, how do the veg-in accessions work commercially? If a breeder uses genes from one in a new commercial variety, does the veg-in Warwick Harper Adams retain some of the IP rights?
So everything in veg-in is open access. So anybody is able to access our information and utilise the results that we've generated. No, we don't retain IP rights. That's it. No, we don't. We do like to be informed of what material you've used and what it's gone on for, because that's really useful for us in future work and obtaining new funding to continue this kind of work. If we can prove that it's useful to industry, that really helps us. But no, we don't retain any of the IP there.
Thank you. Right, we're on to alliums now. So Andy, again, also interested in onion thrips resistance screening. Is this something underway already or something planned for the future?
This is planned. Sadly, no results on that yet, but hopefully in the future.
Great. And then now we're on to, this is Ningwen. Very interesting work. Thank you, Lauren. Can you say something more on the onion drought and flooding experiment? Did you find interesting diversity of the tested materials?
So this work was carried out by Harper Adams, so I'm afraid I don't know any specifics. I do know, again, because we have this diversity set and it does segregate for pretty much every trait, that we definitely identified lines that were more tolerant or less tolerant to those extreme climate conditions. I'm afraid I couldn't give you more specifics than that, but I know that we've definitely identified tolerant lines within our onion diversity set.
Great, thank you. And I think this is probably the final question, and anyway, we're running out of time. So again, from Andy, final question on willow carrot aphid. Were winged aphids used in all the screens?
Winged aphids were counted in the field assay and were used in all the choice experiments. Unfortunately, we weren't able to generate quite enough winged aphids. They're quite a tricky culture to handle, so we weren't able to generate quite enough winged aphids for the control environment full resistance screen.
Great, thank you. Right, I think we've gone through all the questions. So yeah, I think so. Right, so it only remains for me to say thank you very much indeed to our three speakers, Justin, Eleri and Lauren. Many thanks for your time and your really interesting presentations. And thank you to everybody for joining us and for the very interesting questions. And hope to see you at another webinar in the future. Thank you all.